All right, so we're now going to walk through different, not every single option, but different processes or methods that are used to control microbes. So we have physical methods. Physical methods are going to be heat or radiation. And when using heat to destroy a microbe, we are typically affecting protein function. And the heat that can be used is either dry heat or moist heat, and will result in different types of outcomes. So we can sterilize things, or we can also disinfect them, just depending on the method. Radiation, we can have ionizing or non-ionizing radiation. So UV radiation will lead to disinfection. Ionizing radiation is something a little bit more heavy duty, which is going to disrupt DNA and cellular level components by X-rays or gamma rays, right? And that will lead to sterilization. We can also use chemical agents, so we can use gases or liquid chemicals, and these can be used on both animate or inanimate surfaces. And again, just depending on, what kind of surface you have, determines what you're going to choose to use of any of these physical, chemical, or mechanical methods of removal. And then the mechanical methods of removal, that's simply filtration and of either air or liquids, and that leads to just decontamination. All right, so let's kind of go through in detail and walk through some of these different things. We're going to start with physical agents. So physical agents of control are heat and radiation. So heat as an agent of control can be either moist heat or dry heat. And typically when we're using moist heat, you're going to be either boiling something, or you're going to be using steam and pressure in order to have temperatures that are above boiling and expose an object to that high temperature for a certain amount of time. When we use steam, what happens is that you can have really hot water vapor that will cling to different surfaces of different shapes and kill things that are on the surface. When we're using dry heat, we're incinerating things, right? So we could be lighting them on fire, or we've been using our Bunsen burner to sterilize. So we can have really high temperatures and expose and kill organisms on surfaces. But, keep in mind that you can't necessarily, light everything on fire, right? So we're always thinking about what is the method of control that you're going to choose dependent on the type of object. So yes, if we want to sterilize a loop, right, we can put it straight into the Bunsen burner. It's metal. It can withstand that flame. Right? If we put something into an autoclave and it's going to be put under a lot of pressure, you have to make sure you have certain kinds of equipment that are going to be able to deal with the type of pressure. Not everything can be autoclaved, right? We can put things into small little ovens and heat them up and just use dry heat, right? So the type of physical control or method that you're going to use of heat is really dependent on the type of object that you are trying to remove the microbes from. So one of our best methods of, and what we use in the lab a lot, is using steam under pressure using autoclave. It's essentially a pressure cooker, right? You put a little bit of water in there, you seal it up, you add pressure in, you add heat in, and like I said, it creates water vapor that is a higher temperature, and that water vapor then can cling onto the surfaces of different objects in there, whether they're liquids or solids, and can kill off the microbes that are growing in possibly the liquid or cling to the surface of solid objects. So a really good way of sterilizing things. Another method of dry heat is using infrared radiation. So this is an infrared incinerator. It's putting a loop into and exposing it to infrared radiation, which will which will remove microbes from that loop in that way. So again, it's not everything we use can we just light on fire with a Bunsen burner. So if we're using heat in order to kill off microbes from different surfaces, it sort of begs the question, can we go the other way? Can we use cold, or can we remove moisture, right? We talked about using heat and moisture to kill things, right? So can we do the opposite? Can we remove water? Can we make things cold? Yes, but typically this is not going to result in the killing of any microbes. This is just going to be more of a bacteriostatic process, slowing the growth. So yes, if we put things in our refrigerator, if we freeze things, right, it's going to slow the growth of bacteria and prolong the spoiling of the food. So desiccation is the term for drying something out. Yes, we can dehydrate and reduce the ability for bacteria to spoil food by dehydrating certain food. Right, but more of a bacteriostatic process, like I said, and less of a bactericidal process. All right, another method of physical control is using radiation. So using different wavelengths of electromagnetic light or electromagnetic waves in order to remove microbes. So you can use UV sterilization, or you can use, and I say UV sterilization because there's different methods of, it doesn't really sterilize, but that's what it's called, is a UV sterilizer. You can use X-rays and gamma rays. So UV radiation is used quite often in doctor's office, dental office, even in like you go to the nail salon, right? So we have a UV sterilizing cabinet in the lab where we put the put goggles or glasses that you would wear in the lab, and you turn it on and it exposes those objects to UV light, which, removes some of the bacteria from those surfaces and other microbes. It's also used to help us remove contaminants from water and bacterial from water. So I used to work at a pet store, so we used to sell these UV sterilizing for like fish tanks. So essentially what it does is it runs the water past UV light and it will kill off of the microbes or the algae, the smaller organisms that are growing inside a house to keep your fish tank clean. Right. This is showing sort of a larger scale used in like wastewater treatment plants, right, where we're running water past a UV light in order to remove microbes. Now, on the higher end, UV radiation can potentially be hazardous, but it's non-ionizing radiation. It is not as dangerous as using X-rays or gamma rays. X-rays and gamma rays, when we are exposed to them, can potentially damage our cells as well. But you can use X-rays and gamma rays, and those will sterilize and kill organisms on surface, but you also have to be very careful about using them, around people, because it can potentially be cancer causing. All right, so we can also decontaminate by using filtration. So by running water or air through filters, we can have different pore sizes that are small enough in order to filter out certain types of microbes. Right now, we're sort of going through lots of discussions about that and uses of mass and types of mass, and what type of pore size is necessary in order to remove microbes that are in droplets or microbes that are in the air. Right, so there are lots of different levels of filter technology and pore sizes so that we can remove anything that's as small as a very small virus to larger organisms, right? And so this is a process of decontamination, removing those microbes through water or liquids or through the air. Right. There's other sorts of common filters too. So we've talked, we think a lot about masks these days, using water filters in your sink. But there's also air purifier filters, things that you can hook up into your house. There's very advanced filters that they use in sensitive, locations in the hospital, like in the air ventilation system, like where are they going to allow air to come directly into the ICU or into, surgical rooms where you have the most sensitive patients. So there's different ways in which, we're controlling the flow of air also around the hospital. Okay, so we've talked about physical agents, we've talked about mechanical removal, now I'm going to talk about some different chemical agents. So there's lots of different kinds of chemical agents. There's something called a phenol coefficient, which is just sort of a standard of measurement where we compare different chemicals and their antimicrobial ability, right? So if you have a really high phenol coefficient, you're considered like a poison. In lower concentrations, you're just like inactivating enzymes, so less dangerous, right? So different types of disinfectant chemicals that you're probably familiar with. We're just going to talk about a few of them. So alcohols can be used to destroy microbial life. Ethyl and isopropyl alcohols are good for microbial control. They are disrupting membranes. They can also denature proteins. They also desiccate cells. So they're going to destroy most microbial life. They're not very effective on destroying bacterial spores. And they can work on viruses. Specifically, they're better at inactivating envelope viruses because remember, envelope viruses have sort of a membrane that they've taken from a host. They're more effective because of their mode of action than non-enveloped viruses. Hydrogen peroxide can be used. Hydrogen peroxide is a reactive oxygen. We've talked about that throughout the semester, and can be very reactive and highly toxic to cells. And in high concentrations, it can kill spores. In lower concentrations, it's good on bacteria, viruses, and fungi. Detergents, cleaners. So detergents are surfactants. So I mentioned a surfactant in the last video, and a surfactant is a molecule that embeds itself into and sort of replaces a phospholipid in the phospholipid bilayer. And eventually, after we've inserted more and more surfactants into the membrane, it makes the membrane unstable. So surfactants or detergents that are positively charged tend to be more effective because they bind to that negative charge on the surface of the bacterial membrane. Heavy metals or any compounds that contain heavy metals like mercury, gold, silver, copper, they tend to inactivate proteins, so they bind to proteins and shut them down. The drawbacks to using heavy metals is they build up in the ecosystem. So eventually they can, build up within the human body. They can be toxic to humans. A lot of people have allergic reactions to them. Some of them are precious metals that are really expensive, gold and silver, not necessarily the most practical or cost-effective way in order to control microbes, but they do and can prevent the function of microbial growth and proteins. So I just wanted to show this because this is going to lead into what we're going to see in the lab portion. So this is a plate that has two heavy metals sitting on it, and we can see that this plate has been inoculated with bacteria, and there is a ring around the metal, which is preventing microbial growth, right? It's a zone where there is no growth. And then the further you go away from the metal, we start to see more and more growth, right? So this is what we're going to observe when we use different chemical agents in the lab. We're going to put them onto a plate and observe the growth patterns and see if those chemicals inhibit the growth on a plate. Then we'll also move into doing a similar technique for looking at antibiotics. All right, the last chemicals I'm going to talk about are acids and alkalis. So changing the pH is going to also disrupt a protein function, right? So using people clean with like vinegar, even lemon juice, things like that. But they can tend to be caustic and corrosive. So again, thinking about what kind of surface, like what you're trying to remove. All right, so that is an overview of physical, chemical, and mechanical methods of removing microbes. We'll do the lab, and then we will move into talking about the function of antibiotics.